Hello, everyone, and welcome to today's keynote webinar titled Rational and Combinatorial Design of Peptides for ss-mRNA and ds-mRNA Separation and Purification. This webinar is part of the 7th Bioprocessing Virtual Event Series. I'm Matt Thorne of LabRoots, and I'll be your moderator for today's event. The educational web seminar is presented by LabRoots. Before we begin, I'd like to remind everybody that the event is interactive, and we encourage you to first participate by communicating with other attendees using the live chat feature. You can find the live chat located in the bottom left of your screen. You can also participate by asking as many questions as you'd like during the presentation. To do so, simply type them into the Ask a Question box and you click Submit. We'll try to answer as many questions as we have time at the end of the presentation. If you are having any trouble seeing or hearing the presentation, you can click on the Help Desk button from the lobby within the event. As a reminder, this presentation is educational and offers free continuing education credits. Click on the Continuing Education Credits link located within the abstract on the left of your screen and follow the process to obtain your credits. Now let's go ahead and get started. I'd like to present today's speaker, Pankaj Karande, Associate Professor, Chemical and Biological Engineering at Rensselaer Polytechnic Institute. For a complete biography on our speaker, you can find it also on the left side of your screen. Pankaj, you can now begin your presentation.
Great. Thank you, Matt. Can you hear me well?
Yes, we can.
Perfect. Let me start by saying good morning and good afternoon to our attendees on both sides of the time zone. Thank you for the introduction, Matt. It's a real pleasure to be here and share some of the work that we're doing in my lab with the attendees. I have to say, this is work that we started maybe about a couple of years ago. So this is really fresh out of the oven and preliminary in some ways, but we have some very exciting findings that I would like to share with you. And I hope that I can get you as excited as we are about these findings. Now, obviously, as the title of this talk suggests, one of the things that we are interested in is developing new modalities for purification of mRNA-based vaccines, and that's what I would focus on. I will say, however, that, of course, this work that I'm presenting is applicable, platformable to other types of RNA-based vaccines beyond the ones that I'm going to talk about today. And I'd love to hear some feedback or questions or potential avenues for collaborations where we can bring these technologies that we have been working on, these platforms that we have been developing to help further other technologies. So I don't think it would be hyperbolic to say that the development of mRNA-based therapeutics has been one of the biggest success stories in medicine in our lifetime. Personally, for me, I think the evolution and development of RNA therapeutics is just fascinating. And I understand this is not the forum to discuss that. But what fascinates me really is that it's been 7 decades, right? 70 years almost since the discovery of RNA, and it's taken that long to really transform the potential of this molecule into a very effective therapeutic candidate. And I think there are 2 key developments that happened in less than a decade. One, the appropriate modification of the nucleosides so that this molecule can elicit the right immune response, the appropriate immune response without it being adverse. And then the second, of course, is the encapsulation of this molecule into lipid nanoparticles for its effective delivery, which is at least half the challenge, if not more, in terms of getting the molecules to where they need to go. And so it is not surprising if you look at the growth in this field, especially since 2020, when we had the COVID-19 vaccine. And again, I'm just showing an image here that has the Moderna and the Pfizer vaccine. I'm aware there are more in development or already out in the clinic. But if you look at the overall market, the global market for mRNA vaccines and therapeutics, before COVID, that was around $600 million globally. And after COVID, that has increased by 2,000%, 20 folds to about over $10 billion. And it's projected that by 2050, this is going to go north of $50 billion. So there is a tremendous potential here, commercial, scientific, technological, that we can explore. And that's what really got us interested in working in this field. We are doing this work in collaboration with many other partners who I will describe as we go along. I also want to say to the audience, if you do have questions as I'm going through the talk, please don't hesitate to stop me and ask questions. I'll also leave enough time at the end of the talk to answer any questions that you have. But I think there is one statement that really eloquently summarizes where we are headed. And this is from Melissa Moore, who was the former chief scientific officer at Moderna. And her comment was, a tsunami of new mRNA derived therapeutics will flood the market in the next 5 to 10 years. And we are already starting to see that. If you look at the candidates that are in clinical trials or in the product pipeline, that number has increased substantially, and it will continue to do so in the next decade or so. But given the promise of this technology, it is not without its own challenges, and some of these challenges haven't been comprehensively addressed. There was a rush to get these therapeutics out in the market in light of the pandemic, but I think there are several areas where we can bring in more efficiency, more productivity, better technologies, better platforms. And the one that I'm going to focus on is the challenge in producing mRNA, right? There are two kinds of challenges. One that is process related. So there are process related impurities that you have to deal with when you're making mRNA in an IVT process. But then there are also product related impurities. The product has its own sort of physicochemical characteristics. There are variants, product variants that have to be addressed. And so if your goal really is to make that desired target product, which I'm showing here. I don't know if you can see my cursor, but the purified full-length mRNA, the schematic shown there, you need the 5' cap intact, you need the entire sequence, you need the poly A tail, you need the 3' end. You have to have that entire molecule intact for it to have therapeutic potentials. And there are a lot of impurities that are competing with this molecule, as I mentioned, both from a process standpoint, but also from a product impurity standpoint. And my lab has been working on several of these challenges, how to purify these impurities. And given that we only have limited time today, I'm going to focus today's talk on one specific impurity, which is the formation of a double-stranded RNA in an IVT process where you're trying to make the single-stranded mRNA or single-stranded RNA for other purposes. And the reason that we decided to focus on this impurity, as we are working on others, this one is a critical impurity. There are many, many reasons why double-stranded RNA can form in an IVT process. And again, this is not the forum to go into that, but there are biological reasons, there are process-related reasons why you can have the formation of double-stranded RNA. Either way, it is an undesired impurity for several reasons. First and most important is that it is an undesired byproduct of producing single-stranded RNA because it does have immunogenic properties. And there is a putative hypothesis that the adverse reactions to vaccines, to the COVID vaccine, that some patient cohorts showed had to do with the small amount of impurities, 1 to 4% of double-stranded RNA in the vaccine that could not be separated. So that's one challenge from a clinical outcome standpoint, right? The second, from a process standpoint, this is an impurity. The double-stranded RNA is an impurity that's difficult to remove because it shares many of the same physicochemical properties that define the single strand. And so this is really like trying to find a needle in a haystack. You're trying to take advantage of few subtle differences between the single-stranded and double-stranded. And so it was very clear to us when we were first introduced to this problem, we started thinking about it, that there is obviously a need, a gap for a more facile approach, a more facile way to separate this double-stranded mRNA impurity from the single-stranded mRNA. And if you don't mind, I will just use ds-mRNA and ss-mRNA to abbreviate these two molecules in the rest of the talk. Now, this is a challenge that's not entirely new to us. My group has been working for the last decade and a half on designing ligands to biologics, biologic targets of interest, to be able to separate them in downstream processes. And I won't go into all of the examples, but there are several. These are published. Whoops, let me come back here. I'm sorry. This is going to take me a couple trial and errors to, there we go. But there are several publications that are already out there. I'd be happy to talk about these. But in the past, we have designed ligands, short ligands, peptide ligands against different types of targets, where the interest is in separating them in a downstream processing. Right. I hope you can see the slide that I am on, which has a few of our papers. Matt, could you please confirm that for me?
I'm showing you on slide 7.
Okay, then I should be on slide. Give me, how do I get back?
You can use previous or next to go forward, to go back.
Um, I have pushed to audience for some reason in
Yep.
the green.
So if you need to,
Got it.
there you go. Perfect.
Perfect. Sorry about that, folks. So there we go. As we started thinking about these challenges, I think I'm experiencing the same issue here. You know what? That's totally fine.
I
It's
think
okay.
I think it's just a very slow animation push.
Got it. Got it. Okay. Um, there we go. Okay. So this is our wall of fame. These are all the publications where we have shown that we can design peptides as short ligands to target biologics. And we have looked at uh, antibodies, we've looked at proteins, enzymes, nanobodies, other types of protein constructs. But beyond that, also modifications on that protein backbone. For example, carbohydrates or deamidations. Um, but I have to say that everything that we had done in the past was directed towards proteins. And more importantly, we were designing ligands to target the product of interest, not specifically the impurities. And in this effort, we are actually kind of flipping these two ideas on the head. We are moving away from proteins to a nucleotide based molecule, but also we are focusing on removal of the impurity rather than capturing the target product of interest. So I just wanted to make sure that we are aware that this is a slightly different approach to this problem. So let me tell you a little bit about what those approaches are. The first approach is combinatorial screening, where we take advantage of large libraries. And the second is rational design, or what we call rational design. And the idea here is that you look to nature, you try to find proteins that are known binders to your target of interest, and you learn from them. You look at what the binding domains look like, what is their sequence, what is their composition, what is their chemistry, what is their physical chemistry, stereochemistry, and you try to learn from it. And you try to mimic that, or in simpler words, you try to cheat. You just cheat what nature has done, and you try to see if that will work for your specific application. Now, the advantages of each of these methods are somewhat orthogonal. If you're doing combinatorial screening, you're looking at the benefit of numbers, the power of numbers. We can employ libraries like phage, yeast, E. coli, ribosomal display, and we can test libraries in a single screening that have a diversity of anywhere from 10 to the 6 to 10 to the 11. For the sake of this talk, we're going to focus on phage display, which has a diversity of 10 to the 9. So a really large space of both sequence and compositions. And so the advantage here is that you have a very broad and a randomized search space. You're not introducing any biases up front. You're truly looking at a random library. On the other hand, in contrast, the rational design approach is based on a few maybe a handful of protein templates that you know have evolved in nature to bind to your target, and you try to learn from them. So the libraries here are much, much smaller, substantially smaller, on the order of maybe a few hundreds, if you're lucky, if you find that many templates. And so you are looking at smaller libraries, but you have a very narrow and a very focused search space, right? So that is the advantage, where you know that evolution has already worked, has done the work for you to evolve that composition and sequence for it to have the right binding properties. And then there is a third approach, which I won't spend a whole lot of time discussing, but I'll give you sort of a preview into where we are going with it. But let me set the challenge, right? If you think about a phage library, and that's what I'm trying to show with that gray box there, and you're just looking at a small sequence, maybe about 12 amino acids long, the potential search space, even for a 12 amino acid long peptide, is in the order of 10 to the 15. So even with combinatorial screening, 10 to the 9 might seem like a large number, but you're still screening 1 millionth of that potential sequence and composition space. And then if you look at rational design, which is the little small blue box there, and of course these are not drawn to scale, but that library is much smaller. And so our idea is that we can employ both approaches in tandem. We can learn from them. We see what clues we get as to what kind of amino acids, what sequences we need for good binding, and then perhaps use that understanding to scan the remainder of that space that we otherwise would not have been able to do, or is experimentally expensive and prohibitive. And so we can optimize around the findings of both of these approaches, combinatorial on one side and rational on one side. And so I'll focus on those two strategies and our results from those two approaches, and then maybe spend just a minute or so how we will use that information for in silico optimization. So let me start with combinatorial screening first. I have a sense that some of the folks in the audience might be familiar with this. If you're not, I have sort of one introductory slide here. But what we are doing is we are leveraging a commercially available library that you can buy from New England Biolabs. It's called Ph.D. 12. The PH and D here stand for page display. 12 stands for the length of the peptide sequence. So these are all 12 amino acids long. The diversity of this library is on the order of 10 to the 9. So we start with this phage library and we challenge our target, which in this case, again, keep in mind, we are going after the impurity. It's the double-stranded RNA, the ds-RNA. We immobilize that with a biotin at the 3' end on streptavidin-coated magnetic beads. So we immobilize the target and we challenge it with the soluble phage library. And so phage that have sequences that have an affinity towards the target will bind to the target, whereas the ones that do not have an affinity will stay in the supernatant. And so we remove the supernatant, we save that supernatant. We do want to analyze what doesn't bind, because that still is very important information for us. And then we do a second wash where we remove the phage under stringent wash conditions to remove whatever is bound. Now, keep in mind, that is our library of interest. That's the pool of interest that potentially has candidates that bind to our target, the ds-RNA, with reasonably good affinity. And we amplify that phage. A portion of that amplified library is taken to next generation sequencing because we want to know what those sequences are. And the remainder is employed in a second round of screening. But this completes one round of a phage panning cycle, right? And we have looked for affinity. Now we're going to do another round of phage panning, but this time we are going to introduce one small change. We're going to take the library that we got from the previous screening, and we're going to challenge it with our competitor. And the competitor here is the single-stranded mRNA. We want peptides that bind to double-stranded, but not to single-stranded. And so we pre-incubate the library with single-stranded RNA. That allows us to eliminate phage and those phage-bearing sequences that might have affinity to single-stranded RNA, allows us to eliminate that. And then we continue the screening as I described before. And that forms the second round of screening, where we are now looking not just at affinity, but both selectivity and affinity. And we carry this process for another couple of rounds. And you can keep going, but as you'll see in the data that I show next, four seems to be enough. You start hitting a point of diminishing returns after 4 rounds of screening. So here is what that data looks like. So let me tell you, on the x-axis, we have results from different rounds of screening going from round 1 to round 4. If you remember, in round 1, we are screening for affinity, but beginning from round 2, we are also screening for selectivity. So we have both affinity and selectivity built in into these different rounds of screening. The y-axis shows you the number of unique peptide sequences that we find in the phage pool that looks promising at each round of the screening. And you'll see a dramatic drop in going from round 1 to round 2. Actually, you'll see a dramatic drop in going from the initial library, which was 10 to the 9, and now we have about 10 to the 6 from the first round of screening. So three orders of magnitude reduction. And then once we employ the selectivity criteria, we see another 100-fold reduction. And then another 10-fold reduction in third round of screening, and it starts plateauing off. The y-axis here is showing you the actual numbers of interesting sequences that we are finding at each round of screening. I should have mentioned this before. We do about 450 million reads per round of screening. And this is where next generation sequencing has become a really powerful tool. This, even about a decade ago, if you wanted to do this with Sanger sequencing, where you're limited to maybe 5, 10, 100 at most, this would have simply not been possible. So the introduction of next generation sequencing has really helped make the high throughput screening also a high throughput evaluation. All right, so then moving on, as I mentioned, we are now looking for both stringency, which is our measure for affinity, and then this co-panning with the competitor is allowing us to be very selective, where we are picking candidates that bind one versus the other. How do we verify that we actually have sequences that show good selectivity? We conduct ELISA, and all of these results that I'm going to show you are done at the population level, at the phage pool level. We immobilize either single-stranded RNA or double-stranded RNA, or a blank control that has nothing immobilized. And we take those phage pools from the different rounds of screening, and we incubate them with each of these three targets. And then we do an ELISA with an anti-phage antibody to see how many phage have actually bound to the target. And the figure on the right shows those results from ELISA screening. So in orange are bars that show phage that have bound only to the double-stranded RNA. Blue is phage that have bound to single-stranded RNA, and then gray is just a background control. This is a situation you face in any kind of biological display method, where you're still going to keep pulling binders that bind to your well plates, to your polystyrene, to your background. And so we're always conscious of it, that we want to remove that population. Either way, the central point of this figure is that at the population level, which is the orange bars, we find clones that bind to the double-stranded RNA far better compared to the single-stranded RNA. So this is a sanity check that we do. This gives us the confidence that the pool that we are looking at really contains binders that have both features: good binding to our target of interest, which is the double-stranded RNA, and also binding with good selectivity, which means they are somewhat agnostic, not promiscuous, but somewhat agnostic in terms of binding to the single-stranded RNA. A further sanity check that we perform is shown in the data here on the next slide. And what I'm showing here is, if I take 5 top peptides, top performing peptides, and the measure here for good performance is the frequency of occurrence. So if a peptide sequence appears frequently during our sequencing, we associate that with good binding, right? So it is a surrogate measure, keep that in mind, but with our past experience, it has it has shown good correlations between performance and frequency. So on the x-axis, we have frequency of occurrence. On the y-axis, we have frequency of occurrence. On the x-axis, we have identified and labeled 5 top candidates from each round of screening. And as you go from left to right, from round 1 through round 4, you'll see that these sequences are continuously enriched. And so we have further confidence that these sequences actually do show the performance attributes that we are looking for. An even more global way of looking at this data. Now, I'm not picking just the top 5 candidates, but I'm looking at the entire library at each round of screening. And so let me tell you what this figure demonstrates. It's a busy figure. Let me walk you through it. On the x-axis, this is a heat map. On the x-axis, we have numbers going from 1 through 12. They represent the position of a given amino acid in that 12-mer sequence. And on the y-axis, we have 20 rows. Each of the rows is dedicated to a specific amino acid. Of course, I'm not showing you which amino acid is in which row in the interest of protecting these results, but you have 20 rows that show the 20 different amino acids, and then 12 columns that show the position in the sequence where those amino acids belong. And this is data shown for round 1. There is a color code with blue showing the lowest frequency of occurrence and yellow, orange showing the highest frequency of occurrence. And if you remember, frequency of occurrence is a surrogate measure for performance. And so if a spot lights up, that tells us that that specific amino acid at that specific location is important for good and selective binding of the peptide to the target compared to the competitors. And we do this over all 4 rounds of screening. And the first thing you'll notice is that if you look at the results from round 1, they're somewhat diffuse. There's a smear of colors all over. But as you go from round 1 through round 4, we start a little bit more start seeing a little bit more granularity to the results. We start seeing the emergence of hot spots. You'll also notice that the scale on the color legend changes from round 1 through round 4. So we are now starting to see specific amino acids in specific locations enriched. And what that tells us is trends. What kind of trends are we seeing in sequence propensity, in positional preference, and more importantly, is there a periodicity? Remember, these ligands are binding to a molecule that is helical, that has its own periodicity of nucleotides. And so this is not entirely surprising, but being able to point out what that periodicity looks like and for what amino acid really gives us some good design heuristics. Okay, so keep this data in mind. I'm going to shift gears real quick to talk about the complementary approach, which is the rational design approach. And then in a few slides, I'll try and bring these results together just to tell you what we have learned from them, right? So the rational design approach relies on known proteins that bind to RNA, single-stranded as well as double-stranded. And what we do is we look at the binding domain of those proteins, especially when the sequence has been resolved. If structure is resolved, that's an additional advantage. I'll talk about that in just a minute. But we take the binding domain and we literally chop it up. We chop it into short peptides that are 16 amino acids long. And then we create a library where we have overlapping peptides. So we are doing a very fine epitope mapping, if you may, on that entire binding domain. And that gives us a library of sequences of potential candidates that we want to bring further for testing. For this particular work, we had about 6 to 7 different protein templates that we identified from prior literature. And the library size was about 350 candidate peptides, so on the order of a few hundred. All right, we do actually synthesize these peptides in our lab using a high throughput multiplexed peptide synthesizer. These peptides are synthesized on cellulose discs. Once the synthesis is done, we actually dissolve that cellulose disc. We reconstitute it in DMSO and PBS. We print them on microarrays. This is done in a spatially addressable manner, so you know exactly which sequence is at what location on that microarray. These microarrays are then challenged with the labeled target. And we are conducting assays with both labeled single-stranded and labeled double-stranded because we want to try and see the differences in the binding ability of these peptides towards these two molecules. The microarrays are incubated with the labeled target. We wash them, we image them. And the idea here is that if we can isolate or extract the fluorescence intensity at each peptide spot, we can correlate that fluorescence intensity to the amount of binding of RNA to that particular peptide. I'm not going to show you any microarray images, but I'm going to show you the analysis from those microarray experiments. And so again, on the left-hand side, you're seeing a plot. On the x-axis, you'll see numbers going from 0 to 350. Those are the 350 peptides that were part of this library. Each peptide has been assigned a random index uh, from 1 to 350. On the y-axis is the intensity of RNA, whether single-stranded or double-stranded, bound to that particular peptide on the microarray. In blue, I'm showing the intensity of single-stranded RNA bound, and in orange is the intensity of the double-stranded RNA bound. And right off the bat, you'll notice that there is a huge scatter in both these clusters. There are several points here in blue that are higher than those on the compared to the orange ones, and vice versa. Another way of looking at the same data is instead of plotting the actual intensities, I'm plotting a rank order. And what I mean by that is the higher the rank order, right, going again from 1 to 350, better is the binding. So on x-axis, you have rank order for double-stranded RNA. On the y-axis, you have rank order for single-stranded RNA, right? Now, of course, the points that lie on the diagonal or close to the diagonal, those are peptide candidates that seem to be promiscuous. They bind to both. They don't show selectivity, right? Further ahead on the left top region of this plot, better is the binding. So we are trying to identify peptides that have both good binding and good selectivity. And that's what those two ovals are showing. The one in red at the bottom of that plot, those are peptides that are lead candidates for binding double-stranded mRNA selectively. And those are the ones we are interested in for this particular discussion because we want to remove the impurities. However, the peptides that are in the black dotted oval on the top left of this plot, they too are interesting because those are candidates that bind to single-stranded RNA selectively. And so you can think of a different process where instead of trying to capture the impurity, you're actually trying to capture the product. And in that case, these would become relevant. So we could go either way, but I'm again going to focus on using the double-stranded RNA specific peptides as ligands for this work. Now, one of the things that we were curious about is now that we have leads that we've identified from rational design and leads from the combinatorial screening, we wanted to see if there are any similarities between them. Right off the bat, one thing we noticed that when we were designing peptides from naturally occurring protein templates, we saw a high abundance of alpha helical structures or alpha helical motifs in those binding domains. And we were curious, and this is not an observation that we made, but it has been made by folks before us as well. And we wanted to see if the combinatorial library screening also gave us peptides that had a propensity towards these alpha helical structures. And I'm showing you the structures of the top 10 peptides that we isolated from combinatorial screening. And you'll notice that 80% of those, 8 out of 10, have some to complete alpha helical structure. There are 2 beta sheet peptides or relatively unstructured peptides, I should say, that also look interesting. And at this point, what we are trying to do is take all of this information from the sequence and composition trends that we see from combinatorial screening, the structure, sequence, and composition trends that we see from rational design, and then combine that information to go to in silico optimization, where we are trying to dock these peptides, model these peptides, understand the fundamental binding interactions, and then taking leads from docking to full MD simulations. That is work that is still in progress, and I would love to share with you perhaps at another venue once it's fully done. But we are seeing some exciting results where we can start saying something intelligent and insightful about how sequence, composition, structure of a short peptide ligand all come together for it to be an effective binder to an RNA molecule, whether it's single stranded or double stranded. All right, so then moving on, I motivated this work by saying that we do need a good downstream purification separation process, and we're looking at ligands that can be employed in those processes. So obviously we have to check whether these ligands work. Everything I showed you was in the context of phage or microarray, and that's not process relevant. What is process relevant is to put these ligands on appropriate supports and test if they actually perform well in process relevant conditions. And when we start thinking about supports, you have several options. You have your porous affinity beads, your gel filtration, you have other kinds of chromatographic approaches. We work on two-phase affinity precipitation approaches as well. And we could spend the whole day thinking about which one is the right support. That's not really the goal here. The goal is to show that what we have discovered are valid ligands. And so we decided to go with membranes, grafting these peptides on membranes. And there was some thought behind it. Um, when you are looking at RNA, whether it's single stranded or double stranded, you're really looking at very, very, very large molecules, which in many conventional chromatographic formats may be limited by their diffusion. And so convection seems to be the big driving force here, where membranes have a significant advantage. And so that's the reason why we chose membranes as the appropriate supports to test these peptides on. If you have questions about this, I would love to discuss this at the end of the talk. Um, but here's what we did. We took one of our lead candidates. I'm going to call that candidate P312. P312 is a peptide that's selective towards double-stranded RNA, binds to double-stranded RNA with good affinity, and is somewhat agnostic to single-stranded RNA. We grafted these peptides on modified cellulose supports. And another interesting thing that happened during the course of this work is we serendipitously discovered that we can actually graft these peptides on off-the-shelf filter papers. So no fancy membranes, these are still cellulose, um, but we can actually graft these peptides. Even better, we can actually use conventional solid phase peptide synthesis approaches and directly build the peptides on these supports. So the data I'm now showing you is of peptides grafted on cellulose-based filter paper membranes, which we are using as supports. Um, I'm showing you two chromatograms here in the figure in the center. The red one is for an injection of a 10 nanogram per microliter single-stranded mRNA at a flow rate of 0.2 mils per minute at a total injection volume of 1 mil. Likewise, in black is the chromatogram that's for 10 nanograms per microliter of double-stranded mRNA, also at 1 mil total injection volume and also at a flow rate of 0.2 mils per minute. And what's interesting to note here is that if you look at the elution, you'll see that the red curve basically plateaus off. Any inflection that you see in the curve is just because of buffer changes, right? So you can think of that as being the baseline for the black curve. But the black curve, which is your double-stranded mRNA, um, shows a pretty high elution peak. In simple words, that tells me that in a flow-through process, the peptides grafted on these cellulose membranes were able to bind to and hold down the double-stranded impurity while allowing the single-stranded RNA to go through, which is exactly what our goal was when we started the discovery process. A more quantitative look at the same data is shown in this table here. Let me walk you through what the different columns are. The first one just tells you what the feedstock is. The second one tells you how much mass in terms of nanomoles we've injected of each of these, single-stranded and double-stranded. Um, the bound nanomoles tells us how much actually is bound to the membrane. Eluted tells you how much we can actually remove from that membrane. And that's important because if you want to think about a support that you're going to use over and over again in a process relevant format, you want to have good elution so that you can take that membrane, clean it, bring it back, and reuse it again. And then the final column is just how much of the bound mass or bound moles can we recover. And so two key numbers here. The first one is the ratio of double-stranded RNA binding to the peptides compared to single-stranded. We have about an order of magnitude difference. Um, we can certainly improve upon this, and we do have candidates that show better performance now. But that's the kind of difference in binding that we need to see to remove the double-stranded RNA impurity. The other one is 92% bound moles recovered. So most of what we are binding, double-stranded RNA that we are binding to the membrane, more than 90% of it can be recovered in an elution step. We are not showing you results here on cleaning, but we are also looking at cleaning with NaOH to remove whatever else is bound or impurities that we can then bring the membrane back to another cycle of operation. So reusability and cycle life. I should mention that these experiments were conducted with amounts injected that were far beyond the saturation capacity. So there is still some optimization to be done to get the exact process conditions right. But this to us sounds and looks very interesting. So I'm nearing the time I have for this talk. So let me just maybe make a few comments on what we have found. We have used two approaches, very complementary, very orthogonal. One is rational design based, trying to mimic nature, other completely random and combinatorial. We found good leads from both of them. We have observed distinct amino acid compositions, their propensities, their periodicities in the sequence, which can further inform how to optimize and improve upon the performance of these sequences. We found certain structural elements that seem to be conserved, especially the alpha helical structures that make the peptides bind really well to these RNA molecules. I spoke briefly about the ongoing efforts. We're looking at docking, molecular modeling, but then also more refined experiments with SPR, ITC, fluorescence polarization to get better quantitation of the binding metrics, the KD, the Bmax, so on and so forth. And finally, I hope the impact this work will have would go beyond the molecules that we've studied. We've looked at mRNA here, but we are already working on applying these approaches and these methods to other targets, including DNA, circular RNA, RNA with other types of modifications. And so we are hungry to extend these methods, extend these platforms that we have developed to other kinds of candidates in this space, in the nucleotide based space. And so if you have um, suggestions, or if you want to chat about potential partnerships or collaborations where we can maybe bring these approaches to solve the challenges that you have, I'm always ears always eager to hear more about it. And then finally, let me thank the people who actually did the work, so I get to talk about it. On the top left, there is Claudia, who led all of the discovery work, the screening work on peptides. Next to her is Riddhi. She's the in-house expert on grafting things to membranes, peptides, other kinds of molecules. Next to her is Thomas. Thomas is our in-house expert on doing all the static dynamic testing of these peptide grafted membranes in process relevant conditions. And to the far right is my colleague, Dr. George Belford, who is an expert in membranes, has been working in this field for the last 40 some years. This has been a great collaboration where we do the discoveries, we identify the molecules, the peptides, but then we combine it with the right kind of membrane supports to integrate it all in a single module that's relevant for a downstream separation process. Of course, there are other folks who helped us. We at RPI are lucky to have fantastic cores. So I want to thank all the core directors who've helped us. I do want to thank other members of the group who've been also helpful in different aspects of this project. And then, of course, Moderna, who funded this work. And finally, to Lab Roots, all the attendees, thank you for time and attention, and I'd be happy to answer questions if any.
All right, excellent. So thank you very much for that great presentation. We're going to jump into the live Q&A. So if you do have any questions that you haven't submitted, you can go ahead and do so in the Q&A box on the bottom right there of the screen. So let's go ahead and get started. First question I see here is, can this approach be applied to other RNA-based targets?
Certainly, and I think I alluded to this in my conclusion slides. At the end of the day, you get what you screen for, and I think the message here, if I may underscore, is that by by crafting the right search process, and by that I mean, the way we conducted the phage screening, the way we use the combination of combinatorial and rational design approaches, we've successfully used them in the past on other protein-based targets, proteins of all kinds of flavors. And so I certainly think that this can be extended to other types of RNA-based targets, which include circular RNA or RNA with specific regions that need to be targeted. But yes.
All right, thank you. Next one I see is, how stable are the peptides when conjugated
Conjugated to the membranes?
Conjugated to the membranes, yes.
Sure. So there are a couple of flavors, right, here that we could discuss. So the stability of the peptides on one hand could depend on, for example, proteases or other leftover protein-based materials in the IVT that could potentially cleave the peptides. We always screen the peptide sequence against proteolytic sites, and we try to avoid, so that ensures that the peptides would be stable to proteolytic activity. The other question about stability is, if we do the regeneration of these membranes and the wash of these membranes at one normal NaOH or conditions similar to that, how stable would it be? Our preliminary studies indicate that they are stable. Of course, we have to do more comprehensive studies, but at this point, they seem to be fairly stable. So no proteolytic cleavage or um, no deconjugation from the membrane surface after grafting.
All right, next one is, have you looked at different lengths of RNA?
Yes. So for the sake of these studies, we chose poly-IC as a model for the double-stranded RNA. And the simple reason being, it's well characterized. We have some idea of its structure. And we did look at two different molecular weight distributions of poly-IC, high molecular weight and low molecular weight. One of the challenges of working in this field is you don't have a single molecular weight distribution, or single molecular weight, you always have a distribution that you have to work with. But we were actually pleasantly surprised that our candidates actually showed good performance against low molecular weight as well as high molecular weight poly-IC. So that's encouraging. But is that comprehensive? Probably not. We would actually have to look at the entire possible distribution in the IVT context, but this is certainly promising that our peptides seem to be somewhat, at least the lead peptides that we have tested.
All right, looks like we've got time for one or so more. How does the structure of the mRNA therapeutic affect the peptide's affinity to the ds-RNA contaminant?
Yeah, that's actually a really good question, and one that we are trying to sink our teeth in now. Now, of course, this is going to be context dependent, right? Because there is always going to be this kind of this tangle, if you may, between what the single-stranded RNA is and what kind of double-stranded RNAs are possible. So definitely a sequence-dependent question. But I do think that the structure will play a big role. Now, I didn't get a chance to talk about this, but we are submitting this manuscript soon, and we actually have shown how the peptide binds to the single-stranded RNA and how that mode of binding is actually different for a double-stranded RNA. There are specific physical features of the double-stranded that the peptide exploits by wanting to sit in a certain groove, which it can't find in the single-stranded. So we have some ideas of what the molecular mechanisms are and what the modes of interaction look like, at least in the context of the molecules that we have studied. But I would say structure affects this binding for sure. How much kind of starts becoming a little bit of a case specific question. I hope that answer was satisfactory.
All right. So thank you very much again for your time and your important research. Before we go, I'd like to thank the audience for joining us today and their questions. Questions that we didn't have time for, or those that do get submitted during the on-demand period, will be addressed by the speaker via the contact information you provided when you registered. This webcast can be viewed on demand for a year until April 2nd of 2026. LabRoots will alert you via email when it's available for replay, and we do encourage you to share that email with any colleagues who might have missed the live event today. So until next time, thank you very much and goodbye.
Thank you.